Welcome everyone to this week's seminar. Today we have David Ding and Ada Shaw. So David is a postdoctoral fellow at UC Berkeley who obtained his PhD in protein evolution at Harvard and MIT. He is interested in developing high throughput and computational methods to engineer biological systems to address sustainability goals. Ada is an applied mathematics PhD student in Deborah Marks's lab at Harvard Medical School. Her research focus is on developing probabilistic models to predict mutation effects from protein sequence and structure. Broadly, she is interested in applying her skills to therapeutics and sustainability. She will be defending her doctoral thesis in September 2023. So David and Ada.
Hi, everyone. Thanks for the introduction, Jianyu, and nice to meet you all. We'll share some results on protein design with you. And I hope you can see my full screen presentation. Yeah, great. Jianyu, maybe I'll let you admit people into the Zoom room, mostly. Great. Okay, so we'll tell you a bit about what we found trying to design proteins. And the core question that we're asking is how you can genetically tune protein function, i.e., which mutations do you want to put into proteins to change them for different functions, for a tuning of its function. And so this problem can be complicated if you have a lot of epistasis. So on one hand, on the left case, you see an example for a length 2 sequence that can take mutations at the site A or the second site B. And you can see that if you go from a small b to a big B mutation, that effect is independent on which context you have in the other sequence positions. On the other hand, if you have a lot of specific dependencies between different positions, you can see that we'll call this epistasis. You can see that introducing a small b to big B mutation is crucially dependent on the right-hand side the effect of that is crucially dependent on what you have in the other position. And also, let me just check. You can see my cursor, right? I'm not just waving around stuff. Okay, great. So in a higher dimensional space, we often conceptualize this as having a smooth or a very rugged landscape. And you can imagine for sequence optimization, if you take mutational steps and selection, pushing you towards higher fitness. In one case, you can climb to the global optimum. In another case, you might start in a particular, say, valley, climb up some hill through selection, and get stuck in this local minimum. So we know a bit about this problem by looking at models that we've built that learn from sequence. So it turns out that when you, for example, learn from the sequence alignment, that as you increase the complexity, the capacity of these models to model higher order dependencies, you get an increase in performance. So for example, if you look at the sequence alignment and just look at the residue occurrence frequency at a particular position, that model does less well than a model that can consider arbitrary pairwise dependencies between two residues. And those do less well than models that can consider even higher order dependencies. And so, for example, as an example, I'm just showing you results from Adam Rieselman, who's shown that a variational autoencoder that can capture higher order dependencies outperforms pairwise, and those outperform an independent model when learning from sequence. But you can see that there's a trade-off, right? Like in terms of the capacity that these models can use and the number of parameters that are required to model them. So this is also an question of which dependencies do you really need to model biological sequences for their function. So the approach that we decided to take is we're trying to first examine how complicated are these measured biological fitness landscapes, and then later use that information to inform what kinds of models or functions we want to build to model these sequence dependencies. So I'm going to try and convince you here of two parts. The first part is the supervised approach, where we look at experimental combinatorial variant effect measurements. And here I'm trying to convince you that really just knowing the residue mutation preferences are sufficient to predict the combinatorial mutations that we observe in experimental datasets. And then we use that insight to devise an unsupervised strategy. We don't look at these variant effect measurements to learn these mutation preferences from the structure. And we show that these structure-based, local structure-based mutation preferences can actually outperform high-capacity autoregressive methods for designing sequences. So let's look at the first part first. We started by focusing on my favorite protein interaction system, which is a toxin and antitoxin. It's not just that I spent many years working on this, but also I think the power is that we can make high throughput in vivo measurements of these proteins in the physiological context. And so what this looks like is we can generate a random library, say at 10 positions in the antitoxin, and transform these individual library members into individual cells that contain the wild-type toxin. And only those cells that contain an antitoxin variant that is able to bind and neutralize the toxin within that cell, only those cells can grow, right? So over time, we can sample before and after, deep sequence to count the frequency of each of these variants before and after selection, and then get an estimate for how well this antitoxin variant works in neutralizing the toxin, measured by the growth rate of that particular cell. So when we do this experiment, you can see in gray the distribution of fitness effects for all these 10-position randomized antitoxin variants. And many of them don't function at all, similarly to the antitoxin variants that have stop codons inserted into them in red or orange. And we get a good separation from the synonymous wild-type antitoxin variants that don't change, that change the codons, but right, not the antitoxin protein sequence. When we do this experiment twice, we get pretty good reproducibility between the replicates. And you can see that, there's a ball of dead variants here. And that's just because, for one antitoxin variant that doesn't function, doesn't bind the toxin, it goes from 50 reads to 0 reads, and another one goes from 100 to 0 reads, right? So we cannot resolve that. And another way to show you the same data is stratified by the number of mutations that each antitoxin variant has. And you can see, as you would expect, as you introduce more mutations, more of the antitoxin variants don't function as well. But what's interesting is that you can, for example, estimate the fraction of antitoxin variants at a particular mutation distance that pass, say, an arbitrary threshold of half maximal neutralization of the toxin, and then multiply this by the number of total possible antitoxin variants at this mutation distance. This can give you an estimate of like how many total functional antitoxin variants there are that achieve half maximal neutralization across these 10 positions. Okay, so then let's go back to our initial question, which is how complicated is this combinatorial landscape? And the first question we asked is, can we fit just a pair residue mutation effect model to explain these to explain the combinatorial variants without any specific dependencies between mutations? So in other words, can we learn a function that takes just a 20 times n, where n is the number of mutated residues, to explain up to 20 to the power of n combinatorial variants? And so we actually looked at 2 data sets, one that is already existing, where we have a combinatorially exhaustive and complete library among 3 positions in the antitoxin. And we tried the simplest thing with this pair residue mutation effects, which is a linear regression. You can see that it fits pretty well, but there are some very biased residuals, right? It's really not what you, it's really not like solving the entire problem. The next thing we then tried was, okay, well, what if we take this mutation effect matrix that's just 20 times n that we infer and pass it through a logistic, pass it through a sigmoid function, and it's very similar to a logistic regression model. And what this looks like is we, for particular combinatorial variants, say at 3 positions, we learn each individual variant's effect, sum it up, right, additively, and then pass it through this nonlinear transformation to get a fitted growth rate. And when you do that, you can see that for the 3-position library, we can basically explain all of the combinatorial 8,000 variant effects with this site-wise model. In the case of the 10-position antitoxin library, it also improves the fit, although there are some deviations. And it's possible that this fit, right, there's also differences in the reproducibility, the experimental noise in these two different data sets. But it's possible that the fit is worse because neighboring residues are mutated, right? We have chosen to mutate residues that are quite close to each other. Anyhow, this is just like a prelude for later. But we were able to basically explain in these, 10,000, 8,000, or 10,000 observations with a much fewer number of just per residue mutation effect parameters. And so, a natural question is to ask, do you really need all these 8,000 variant effect measurements to learn these 60 mutation preference parameters? So what we did is just subsample those random, those observations we have randomly and train this logistic regression model on it to predict the held out remaining variants. And when we do that, you can see that as we subsample down for the 3-position antitoxin library down to like say 200 random observations, we basically get an almost perfect prediction of the remaining 7,800 combinatorial variants, suggesting that you really don't need to do this very measure all those 8,000 variants. And similar in the 10-position antitoxin library, when you have around 1,000 observations, random observations, you can get a test set Pearson correlation of above 0.8. And this is better than any unsupervised models can give you at this point, at least. Another question that you could then ask is, well, can these per residue mutation effect models work in other proteins, folds, and functions and residues? Some people have showed this before for the individual proteins. Here we collated 5 of them and ran our logistic regression model on it. And you can see that really, indeed, you can explain, 80 to 90% or 95% of the observed combinatorial variants in these combinatorial variant data sets. One caveat is, of course, that these data sets are limited, right? Like many of them don't have that many mutations away from wild type. So, so some of them are limited by only having up to 2 mutations, for example, the GB1 data set. But again, like you could ask, okay, why is the AAV data set, the fit, worse? One is maybe it has more mutations, but another potentiality is that in this data set, you're actually mutating up to 21 mutations among 28 contiguous positions that are very close to each other. So it's possible that they're affecting each other. There are actually dependencies between them. And when you look at these data sets, you also see that we have actually, some of those data sets are amazing because they've measured, like in the GB1 case, 500,000 combinatorial variants. And that's way more than the number of per residue mutation preference parameters that you need, right? The number of parameters is just 20 times the number of residues that are mutated in each of these residues. So we asked the same question again, do you really need all these observations? We'll just randomly subsample them, train this model and the logistic regression model on this, and see how well we'll do in the held out data sets, held out test set. And what you can see is that, oh yeah, we really don't need all this data that we've measured. Quite a small number of them is sufficient. And another way to show you the same data is to normalize the training set size by the number of parameters that the site-wise preference parameters that you need in this residue mutation effect model. And you can see that when you 5-fold oversample that, you can get a test set correlation of that's higher than 0.8, which is higher than any of the unsupervised models can give you. So this was the first part. I'm hoping to convince you that for many combinatorial mutation data sets, just knowing the residue mutation preferences is enough. And that leads us to the second part. And we basically first started off just by asking what really determines these residue mutation preferences that you need to explain the combinatorial variants. And one hint that we get is from a previous data set that we've generated, where we have mutated those 3 antitoxin positions, like in blue, turquoise, and purple, along this like yellow antitoxin. And we've ran all 8,000 combinatorial variants among those 3 positions against 10 different toxin mutant backgrounds that are shown you in spheres here. So what they did was like we inferred those antitoxin sidewise preferences at these 3 positions in the antitoxin from the 8,000 measurements, combinatorial antitoxin measurements in each of the toxin mutant backgrounds and compared them. So for example, if I infer the antitoxin sidewise preferences in the toxin mutant background V5L that does not contact any of those 3 mutated antitoxin positions, those sidewise preferences are almost perfectly correlated to the sidewise preferences that we learn in the wild type toxin case. But in the case where we infer those sidewise preferences in the antitoxin, these 3 positions, in a toxin mutant background that does contact one of those mutated antitoxins, you can see that this particular mutated antitoxin K63, its sidewise preference changes, right? So this is suggesting that maybe not super surprisingly, but that the surrounding residues can change the sidewise preferences. And then we asked, okay, well, is the structural context, is it sufficient to predict the sidewise preferences? And so what we have in one case is these residue mutation preferences that we learned from the combinatorial variant data. And they are very powerful. Those 60 parameters in this particular 3-position antitoxin case are able to explain almost all the variation of the combinations, right? And on the other hand, we can try to learn these from structure, where we use an equivariant graph neural net that's written by Bo and Jing in this case, but you can also use other versions of this to predict the mutation preference. So this graph net is trained on all residues in the PDB, right, weighted and and stratified by folds. And what it takes is the microenvironment around the residue to try and predict which the masked residue should be. So after it's trained and has about a 50% accuracy in predicting masked residues, we can now feed for each of those mutated antitoxin residues the microenvironment around it and use this graph net to give us a mutation preference score. So when we compare these 2 mutation preferences from the experiment versus from the structure inferred, you can see that we get a pretty good recovery of those mutation preferences from structure alone. This is also the case in the 10-position antitoxin library. Here, those 10 times 20 mutation preferences are not as good in explaining all the combinatorial variant effects, but still pretty good, I would say. And if you try to learn them from structure instead of from the experiment, you can recover those relatively well. So that leads us to the unsupervised part of designing combinations. We have a whole bunch of unsupervised models, VAEs, large language models, and structure-based transformers. And here we add this approach using the structures to get the sidewise preferences and then getting combinatorial variant effect predictions just by the sum of those sidewise preferences to get a combinatorial score, and then using very simple Boltzmann sampling that just gives us a handle, a knob to tune the diversity of how we sample from these scores at each positions, and then we get these design sequences. And so from all these models, we can design sequences, and it's usually very expensive, time-consuming to evaluate these sequences, especially because you need to compare models across different temperatures against each other, because there's not a one-to-one map of what temperatures you should use for each model. And but what we realize is that, oh, maybe we can just use a supervised oracle function that is trained on the combinatorial variant data that we have to evaluate those design sequences. So in the case of the 10-position antitoxin library, we can train this logistic regression model on 90% of the combinatorial data, and then try to predict the held out 10%. And this is really an estimate of in-distribution generalization. And you can see you get a pretty good correlation. But you might still say, well, this does not look perfect. Like if it's predicted to be 0.8, then there's still quite a range of actual variant effects that this particular variant could have. So maybe we should really only use this oracle for a more coarse-grained but more robust task, which is classifying whether a particular antitoxin variant achieves half maximal neutralization or not. And when we use it just for this like robust task, it's almost a perfect oracle to classify this. Okay, so then we have this COVs, the combinatorial variant effects from structure. We can design sequences and evaluate them with this oracle. So I'm showing you one example where we sampled sequences at a temperature of 1.5. And the oracle believes that 70% of those sequences fall above this half maximal neutralization threshold. And that's more than the random library has, which only has 20%. Those sequences are also diverse. So there's an average of 6.7 mutations between those sampled sequences, and an average of 5.6 mutations of the sample sequence to the wild type sequence. And there's also diversity at each position where, you could almost expect as the graph neural net believes that at this position L55, you can have a leucine, but you could also have a methionine. At other positions, it believes that, oh, you really need this aspartate or this alanine here. So this is great, but then how can we compare this against other models that sample sequences, design sequences? And to do so, I will summarize this set of sampled sequences just by two numbers. So, in this case, we've sampled, say, 500 sequences, 91 of which are unique. And we're going to summarize this set of sequences at this temperature with two numbers. One is what fraction of those sampled sequences is predicted by the near perfect oracle to be achieving this half maximal neutralization threshold. That's arbitrary, by the way. And how diverse are these designs? Like how many mutations do they have from the wild type sequence? So in this case, we'll plot this dot here where you have a mean of 5.6 mutations from wild type, and about 70% of those sequences function. So now we can do the same thing, sample at different temperatures, sample with different models to get sets of sequences and summarize them as points like this. And the picture that emerges looks like this, where obviously as you increase the temperature, the diversity of sequences increases, but you get less functional sequences. In purple, I'm showing you COVs, which is this independent structure method to choose these mutations. In green and blue, you can see autoregressive models that can learn arbitrary dependencies beyond the structural context. You can actually see that they do worse in generating functional and diverse sequences. And in yellow, you see this method called EV couplings. It's a POTS model that's trained on the sequence alignment for designing and sampling the sequence, and it does even worse. As a baseline, I'm also showing you in dark gray what the random library, the measured random library looks like at each mutation distance. So all these methods do outperform this case, in this antitoxin case, this random antitoxin library. And then in light gray, you see the supervised logistic regression model. So that's really just a way of saying, does the oracle believe that there are more functional and diverse sequences out there? And so there's still a gap that that hopefully future efforts can close. The next thing we ask is, okay, well, can you use this COVs, like combinatorial variant effects from structure method, to generate sequences for other proteins apart from antitoxins? And then it turns out that we can also do this for GFP because we have like a near perfect supervised oracle. So if this oracle is trained on 90% of the data and we evaluate it in 10% held out data, you get a pretty good correlation. There are some bias residuals, and I think that's because in this data set, you would actually want to choose a different nonlinearity, but I can tell you more about this later. Anyhow, but even just using this here, we can then say, okay, let's do the more coarse-grained, more robust task again for the oracle, where we just ask it to classify half maximal neutralization. And it's an almost perfect classifier for that on the 10% test set. And Another way to show you this is like among these test set, the ones that are predicted to be fluorescent to achieve this half maximum fluorescence cutoff, 99% of them do actually achieve it in the experiment. So then we just did the same thing with the different design methods. We sampled at say 10 random positions across GFP. And I should say the GFP dataset is really up to 15 mutations across 230 residues in GFP. So it's different than in the antitoxin case where we've just mutated 10 defined positions. And for each model, if we ask it to design 10 random positions and increase the sampling temperature, you can see that just using the structured microenvironments to design these mutations actually does very well and outperforms both the random library as well as ESMM, inverse folding, and protein MPNN in designing these combinatorial variants. Another game we can play in the GFP case is to do low temperature sampling, but change the number of positions that the model is allowed to change. And you can see that by using this approach, you can actually get even better performance for all models in terms of getting functional diverse sequences. With the Cove's approach still outperforming the autoaggressive counterparts. So yeah, so that's basically it. And so I'm hoping that I've convinced you of two things. One is that the residue mutation preferences are really sufficient to predict a large fraction of the combinatorial mutation effects in observed data sets. I think it does best when mutated residues are non-contacting. But even for contacting or close by residues, we can explain 80% of the observed variance. And then in part two, I was hoping to convince you that the structural microenvironment can give you these mutation preferences. And just with considering those alone, you can actually sample sequences, combinatorial variants that outperform methods that can learn arbitrary dependencies between positions. And so our suggestions for tuning protein functions are these. In the supervised case, where you're able to make variant effect measurements, you should choose preferably n non-contacting residues, maybe just a few of them, and then measure 5 times 20, right? Because we've observed across 7 data sets that if you make 5 times as many observations of the sideways preference parameters, that you can fit a very good nonlinear residue preference model and then make any combinations of your desired function. And in the unsupervised case, where you don't have variant effect measurement abilities, you should also choose non-contacting residues, preferably. And then you can use this course, which is an independent way of looking at different microenvironments to predict the individual residue mutation preferences to get a score for the combinatorial variant and then design sequences. And so our speculation is that, maybe most or all that these high capacity models with up to 1 billion parameters are learning that are trained in sequence alone is really just which residues are close to each other. And so I think there's an outstanding question really, like whether there's really any significant epistasis beyond just the close structural context. There's a couple other observations that I think suggest this, but yeah, ask me if you're interested. And yeah, so thank you for listening to this. And I also am very thankful for, now that I put this slide together, like maybe almost 100 people that supportive people that I've interacted with and learned from, and across 3 different labs. And then in particular, for this work, I would also like to thank Sam Sinai, Chloe Su, and Stefan Paul for their feedback. And we had some funding for this also from the Boehringer Ingelheim Funds. And yeah, any questions?
I have one question.
Sure, whoever you are.
For your unsupervised model, you said you trained it to got like a 50% wild type accuracy.
Mm-hmm.
Why did you choose a model with 50% wild type accuracy? Why not higher?
Yeah, I mean, at the time when we started this, I wasn't aware of any models that have higher wild type sequence recovery. Do you know of any that have better?
Yeah,
wild
so
type score?
I've published one that had 69, and then I had another one, I think I hit 85. And then, but we've also shown that if we like pick models based on their correlation for thermal stability, it's usually best around 60-ish. So
Great, yeah, that sounds awesome. I would love to take a look, and I'll check it out. See whether we can improve that, yeah. Your name's Daniel Diaz, right?
yes.
Yeah, apologies. I mean, I'm probably aware that we're all drowning in a lot of papers, but
No,
yeah,
yeah,
I'll try to take a look.
I
Yeah.
was just curious on like if there was like a reason for it.
Oh, I think I just I mean, for the choice of this geometric vector perceptrons from Bo and Jing was that they had done very careful splits in evaluation of the wild type accuracy. So the training set is split by CAF topology, but also when they train, they make sure that each mutation, like out of the 20, is equally represented. So you don't just get bias in your training, bias from training on a unequal, like shifted training distribution, label distribution.
Yeah, you're talking about these GVP.
Exactly. Exactly.
Yeah.
Do you suggest any way to normalize any target non-specifically growth rate to apply a method? Because as far as I understood, you apply logistic function, which is bound between 0 and 1. So how do I make any target fit to that?
Oh, thanks for the question. What's your name actually? I don't speak. I can't read.
Nikolai.
Nikolai. Okay, nice. Hi, thank you, Nikolai. Yes, I think that's very interesting. In the case of the toxin antitoxins, the sigmoid function works very well, and we've normalized the data between 0 and 1, as in between non-functional antitoxins and wild-type function antitoxins. But for many of the other proteins, what we did is we just rescaled the output of the logistic function that is between 0 and 1 to be an arbitrary range that you learn from the data. And what's interesting is that you actually get certain models, for example, the GFP, I think it just wants to actually have a step function. And what it ends up doing is it just uses the low one curvature part of the sigmoid function. It doesn't use the whole sigmoid function, if you see what I mean. So I think by rescaling and learning those rescaling automatically from the data, you can actually have quite a lot of flexibility in just using a sigmoid function to approximate a lot of combinatorial variant data sets well. But yeah, there's work from Jakub Odwinowski and other people who have chosen to learn more complicated functions like multiple spline functions and so on, that will probably give you a better fit if you're looking into a particular data set and just want to get the best fit of your per residue mutation effects to the combinatorial variants. But I think what's nice about us just using the sigmoid function, a scaled sigmoid function across all these data sets, is that it just reduces, it still does pretty well, and it reduces sort of the decisions that you have to make as someone modeling those effects.
Thank
Does
you.
that make sense? Yeah.
I have two questions. So,
Sure.
my first is that, so you're looking at these residue specific encodings, I guess I'll call them, or I think you called them site independent things. Is it possible that, like, maybe with these evaluation metrics of a Pearson or a
Spearman, that this works well for these data sets because they explain a lot of effects for this evaluation metric. But if you want to use a different evaluation metric, say like substantially better than wild type or something like that, I can't think of it off the top of my head, but like you need more information, then you need something more than just the residue specific information. Like you need these nonlinearities. So the way I would justify it is like, oh, if you have a linear model, a logistic regression, you can get more information, but in order to get like the best performing things, you need these more complex models or something. That's kind of how I've started to justify it in my head. I'm curious if your thoughts on that, if that makes sense.
Yeah, definitely. Yeah, I think it's something we were wondering a lot. So I think I feel like there's two questions that I heard, and correct me if that's not all, but the first one was about whether the evaluation metrics are wrong, and or you could use other evaluation metrics, and in the case of the supervised stuff where we do this like logistic regression, can you see my screen still?
Yep.
Yeah. Right. I think in this case, for example, this is really like the sigma really just makes a lot of sense. It's like it explains almost all your variant effects. And you could, we've plotted the variance explained, the Pearson, the Spearman, right? In this case, I think the Pearson is really justified because it is like a linear correlation. I think what you have to be really careful about with evaluation metrics is when you get to scoring combinatorial variant effects. Like here, you have these unsupervised methods, right? And everyone reports usually just a table without the scatter plots. And you can see that, right, the Spearman correlation, Spearman R, the Pearson R, right, they're all like pretty similar. And like people are like, okay, yeah, like 0.05 increase of our method in terms of Spearman Rs. But the problem is that, maybe you're just interested in like what is the best performing ones. So when you look at this data set here, right, maybe you're just interested like among the best performing ones, can the model predict it? So you're looking just like what you're interested is just for your particular application, maybe whether the model can give you which one is the best, the truly best performing ones, right? So I think the evaluation metrics, they should really be very dependent on your particular application, whether you're interested in tuning like, the entire function here, like going to like, we're interested in like making something that's like half maximally fit. And some people want to do that with antibodies or half maximally good as binding, or whether you're just interested in making the best variants. And then for your second question, in terms of how to get something that's better than wild type, I think the problem is that, I think your measurements can be limiting. So for example, in our case for the antitoxins, that you cannot have a measurement of the antitoxin that is better than wild type antitoxin, right? You cannot resolve that. You can make predictions about it, but then you would have to test that in the background where you have titrated down, for example, the expression level of the wild type antitoxin to fall into this, responsive regime of this transformation, if you see what I mean. And I don't know what it looks like for other data sets, whether, maybe the GFP data set, you cannot measure more fluorescence because, I don't know, the cell might be saturated or something like this. But I think it's something where that's really dependent on the assay measurements that you have.
That makes sense. Yeah, thanks.
Yeah. Oh, by the way, I meant to say that, yeah, there's I think the linked bioarchive and the title and the abstract is pretty old. It's like from last September. It was put up there because I had a fellowship deadline, but we're working right now with all the co-authors on getting a version out that's updated, and we're hoping to put this up on bioarchive like hopefully by the end of this week. So in case anyone's interested in referring back to any of this.
If you chose a non-contacting residues for your recommendations, why would you fit them in a non-linear fashion? Given the results show
So sorry, what
the
I
opposite.
what I meant is it's this result where when in this case we've
It's
chosen
not. I don't think this is the one. I think it was the recommendations from the very end.
yeah, so the recommendations at the very end, and I apologize if that wasn't clear, but what I'm trying to say is that in the supervised case where you can make variant effect measurements, I think this pair residue preference model with a nonlinearity, I think those work the best when you have residues that do actually not have structural dependencies between them. So that's why I'm suggesting you should choose non-contacting residues to tune the pruning function. And then the reason for that is, like, if you look at the 3-position antitoxin library case, right, the combinatorial variance among those 3 non-contacting positions is very, very high, right? It's like you can explain a lot of this variance with just the residue mutation preferences. On the other hand, when you choose these contacting residues, some of which are contacting, you have some variants that deviate from this pair residue mutation preference expectation. And actually, what we've shown previously is like the top, what we and others really have shown previously is like the top deviations from these expectations, those are contacting residues. And you can actually use those combinatorial variants that deviate from this expectation to fold GB1, right? So so it's really suggesting that you get a good fit when you have non-contacting residues that are mutated. But even when you have non-contacting residues mutated, you can still get a relatively good fit, if that makes sense.
Okay, yeah.
And I think what I find exciting about this result with the 5-fold oversampling of these residue mutation preferences is that if you're just interested in being able to predictively tune with mutations the function of a protein, you could just choose a low number of mutated residues, like 3 positions, and then just make 3 times 20 times 5, so 300 observations to get you a very, very good predictive power over unobserved combinatorial variants. And you could do that in a plate reader instead of having to have a high throughput selection system set up. Oh, I don't know if there's any more questions. There's actually one thing that I found very interesting that might be of interest to some of you too, which is, some models learn mutation effects from the sequence alignment. And I think my belief is that learning from sequence alignment can give you some information. You can do a lot of things, but maybe it can also lead you astray sometimes. So in the case of the toxin antitoxin, we actually have 2 different crystal structures of this complex that are homologous. And so they're found both in the sequence alignment that you're training on. But while the overall way that the antitoxin binds the toxin looks very similar, the details are very different. So for example, in this case on the left side, this is the toxin antitoxin that we mutated in our experiments. And you can see that there's this pocket in the toxin. In this pocket, there's an antitoxin that inserts antitoxin tryptophan, like a big hydrophobic residue that inserts into that. And we've shown in previous experiments that this tryptophan is actually really, really crucial, like for binding. This is the one residue that you don't want to mutate if you want to maintain binding between these 2 proteins. But in a homologous structure, right, there is no such cleft. It's just there are more charged interactions. And so you wouldn't really expect the sidewise preferences to be the same for this antitoxin at this position versus that antitoxin at that position. So I think if you just look at, for example, conservation or the frequency of the variant at the position across the sequence alignment, you could be learning a family-wide average that's that can lead you astray for a particular instance of a protein, in our case, the left case that we're interested in. And so, one explanation for why variational autoencoders trained on these sequence alignments are better is that they're learning, that they're like a mixture model, learning the different sort of mutational dependencies for different subfamilies. And I would argue that, like, maybe the best predictor is just the particular structure for your instance of protein that you're interested in. Okay. Any more questions?
I have a question. I hope you can hear me. Nice talk.
Sure.
I just
you.
want to, in terms of the different proteins that you kind of looked at, was there any particular structure motif that may have impacted which protein was more easy to predict? For example, with GFP, you have the beta barrel structure versus looking at this protein you have up here, and it's quite different. Like, was there any particular structure motif that may have impacted the prediction more than the other that you may have noticed from your study?
Yeah, I love this question. It's very interesting, and I would say the 7 protein data sets that we looked at. Maybe Ada, you can talk about this. I think you've looked at these different data sets quite a lot.
Yeah, like as David mentioned, like not necessarily structure would come into play with a lot of these data sets, as like a lot of the way these like mutations were sampled, it was like a lot less likely to draw like contacting residues for a particular mutant. But in particular, yes, for like the AAV, that loop was like very well contacted throughout the sequence and as well as other portions of the sequence. So that was something that we saw in the prediction with a lot more deviations from like the linear model. But yeah, it was quite interesting for a GFP because like with that particular data set, like because a lot of the mutations did not touch each other, it was a linear model was very sufficient to like capture a lot of the mutational effects. Yeah, I don't know if that answers the question.
Yeah, thank you.
Yeah, I also think that we've looked at like 6 different proteins, and we've come up with some explanations of the mutated positions close to each other or not, but this also depends on the experimental reproducibility in each experiment, right? How well we can explain them. And I'm not sure we can really draw very strong conclusions about what types of folds or even tertiary motifs or something like this would be most applicable to modeling like this.
Mm-hmm.
And I should also say, I think there's definitely special sets of residues you can choose that that this per residue mutation effect models will not work at. For example, Frank Poiwick has mutated the 13 active site residues in GFP that form the fluorophore.
Yeah.
And on average, he finds like 5th and 6th order epistatic dependencies, right? It makes sense because you need all these different residues to come together to form this fluorophore. So if you choose like special residues like this or catalytic residues, sets of catalytic residues, you could really see much more dependencies. But on average, if you introduce random mutations across the protein, I think those pair residue mutation preference models do very well.
Yeah. Another example is like the FOQ data set where they looked at like surface contacting residues across like 2 different proteins. Like because there's just so much contact in the mutated regions, it's just really hard to it's not reasonable to use this type of model.
Although I think that remains to be shown. I think we talked about this yesterday. Yeah, we'll see. And then there's also, I think, using structure to predict these residue preferences, I think can also change if you have complete changes in the structure. For example, for GB1, we know that if you introduce, I think, 5 or 7 mutations, you get like a fold switch, right? So, you know, there's no expectation that using the wild type structure from one fold would be predictive of mutation preferences in the other fold, right? So when funny things like this happen, then I would say the bet's off, yeah. But then many proteins don't fold switch, so yeah. All right. Are there any more questions? You can also feel free to email me. I think you can find my email on the Savage Lab website, or Ada's on Debbie Marx's website. Probably should have put them here, but if there's any outstanding questions. And also, if any of you are interested in designing proteins for pH or for sustainability applications, I'm very excited about these topics now, so let me know.
Great. Thank you so much, David, for the presentation and staying around for the questions. And we'll be back with the next seminar in 2 weeks. All right. Thank you, everyone.
Thanks, everyone.